is presented to you by the Chicago Council on Science and Technology in partnership with Carl R. Woest Institute for Genomic Biology. It's sponsored by the Kleinman Center for Genomics in Business and Society. The Carl R. Woest Institute for Genomic Biology, or IGB, is where science meets society, an interdisciplinary institute dedicated to transformative research in technology and life sciences using team-based strategies to tackle grand societal challenges. IGB members conduct groundbreaking genomic research to address issues in the areas of food security, business, and society. The Kleinman Center serves as a platform for enhancing and accelerating the broader impact of the Carl R. Woest Institute for Genomic Biology's research and innovation, building on IGB's innovative genomics research by enhancing its global influence. The center helps IGB provide unique opportunities for economic development, public engagement, and social impact. For those of you that are new to the Chicago Council on Science and Technology, C2ST is a 13-year-old not-for-profit organization that seeks to enhance the public's understanding and appreciation of science and technology and their impact on society. C2ST hosts around 30 free or low-cost programs throughout the year on any and all science and technology topics. We're still focusing on virtual programming now, but stay tuned for a return to in-person programming once it's safe. Please visit c2st.org to learn how you can donate and get information about C2ST's upcoming virtual programs. And remember to sign up for our mailing list so that you can get reminders about these programs as well. Please type c2st2.cnf.io into your web browser and click on today's session to ask questions throughout the talk and evaluate the program at the end. You'll also be able to ask questions wherever you're watching. Our staff will be checking C2S TV YouTube Live as well as Facebook Live for any of your questions. Again, to ask those questions directly, you're going to go to c2st2.cnf.io. Now I'd like to introduce our speakers for today. Dr. Kelsey Witt Dillon got her PhD in evolutionary biology at the University of Illinois at Urbana-Champaign. Currently, she is a postdoctoral researcher at Brown University. She uses ancient DNA and computational methods to explore the history of past populations and is especially interested in domesticated species, examining how the study of ancient domesticated populations can also reveal insights about the ancient human populations they lived alongside. Dr. Ripan Malhi is Richard and Margaret Romano Professor of the University of Illinois at Urbana-Champaign with affiliations in anthropology, School of Integrative Biology, American Indian Studies, and the Carl R. Woest Institute for Genomic Biology. His research areas include using DNA as a tool to learn about evolutionary history in the Americas. And with that, I'm going to hand it over to Ripan.
Great. Thank you for that introduction. Good afternoon, everyone. Today we're going to be talking about ancient DNA, and the talk is going to be split up into two halves. So in the first half, I'm actually going to talk about the history of ancient DNA and the ups and downs in the field. And in the second half, Kelsey is going to talk about two projects that we completed at the IGB that are super interesting and get to the point of doing things that we didn't expect we could do even 5 years ago. So the first question I want to answer is, what is ancient DNA? So ancient DNA is usually DNA that is found in really low concentration and is damaged. And you can find DNA in these contexts in anywhere from a blood stain, a crime scene that's just a few days old, to remains at an archaeological site that can be thousands of years old. So the field of ancient DNA is very young, and so the papers that began to define this field first started to appear in the mid-1980s. And here's one example of one of these papers, DNA sequences from the quagga, an extinct member of the horse family, that was published by Higuchi and colleagues. And so you can see a quagga on the slide from a museum. So the quagga went extinct in South Africa in the late 1800s. And so in the mid-80s, researchers from the University of California and the San Diego Zoo took a tissue sample from this museum specimen and extracted DNA and sequenced a small fragment of DNA. And then they compared this DNA fragment to other DNA fragments in the sequence from living animals today. and then they created a tree of genetic similarity. And what they found is that the quagga is more closely related to the zebra than it is to the cow and the human and other organisms that they compared it to. Now, this makes a lot of sense because the zebra is also a member of the horse family, and the zebra and the quagga have many similar traits in common. So this was a good start for ancient DNA. There were other successes in the early days. For example, there was a study on an ancient giant bird called the moa that was found on Aotearoa, or New Zealand. And when they did the same type of analysis with the moa, they found that it was most closely related to the kiwi that is also found on New Zealand, but much tinier. So that was also a very interesting project. But then ancient DNA researchers began to push boundaries, and there was a group at Brigham Young University who sequenced a dinosaur bone that was 80 million years in age. And so they were able to publish this research in the journal Science, which is the top journal in our field. And so once they published it, other researchers took this DNA sequence from the dinosaur bone and did this, again, this analysis of genetic similarity with other species living today. And you might expect the dinosaur DNA to be most closely related to some kind of bird species who are today's living dinosaurs. But what they've found is that the dinosaur bone was actually most closely related to humans. This is odd, right? So this is exactly what you might expect. This is a case of human contamination, where DNA from a human researcher or someone somewhere in the lab contaminated the dinosaur bone, and the dinosaur bone likely didn't have any DNA preserved in it. And so what you ended up seeing is human contamination that's was mistaken as dinosaur DNA. Right, so that was a low mark for the field. In the early days, there were other researchers who tried to extract DNA from a dinosaur bone. And in this case, they did get a sequence that closely matched birds, in this case, a turkey. But the researchers at the University of Alabama were skeptical because of two things. First, it was a 100% DNA sequence match, and you wouldn't expect 100% match of DNA sequence between a turkey living today and a dinosaur living 65 or more million years ago. And the other reason is because the scientists recalled that they were eating turkey sandwiches in the lab that day. And so they think that this is a case of contamination, not for humans, but from the turkey and the turkey sandwiches that they were actually eating in the lab. Note today that we are not allowed to eat in the labs anymore. Ancient DNA researchers were experiencing contamination in the early days, and they were pretty much paranoid about contamination. And so there were many protocols that were developed that were extensive and stringent to minimize the amount of contamination in a lab and to detect it when it did occur. And so these protocols are with us today. And today at the IGB, we have an ancient DNA clean room where the clean room has multiple protocols and parts of it that prevents DNA. So we have a positive pressure in the room. So when you open the door, air flows out. And so air with contamination cannot flow into the lab. We have filters in the ceiling. So as air flows into the room, it is cleaned. And so you only have clean air entering the room. And we have an anteroom in the lab. So as researchers come in, they have to dress up in these bodysuits that are full covering, masks, hands. So many of you are familiar with masks these days, and booties. And so they can go in and not have to worry as much about contaminating a sample with their own sample or with their own skin or spitting or their own DNA. And so a lot of what we see in this and many other ancient DNA labs is far more stringent than what you might see in a forensic science DNA laboratory. So around 10 years ago, there were some advances in biotechnology, and there were, as a result, rapid advances in ancient DNA analysis and in the projects that were being done. So in the early days, you would just focus on sequencing a small fragment of DNA. In most cases, it was mitochondrial DNA, because mitochondrial DNA is found in many copies in a cell compared to only 2 copies of nuclear DNA. But these days, with these advances, what we can do today is we can sequence hundreds of millions to billions of fragments of DNA. And so you can sequence the complete mitochondrial genome many times over. Or if the DNA preservation is good enough, you can actually sequence nearly the complete genome of a human or any other organism. In humans, it would be nearly over 3 billion base pairs. And so that's pretty impressive and quite a dramatic leap from the early days. The other thing that we could do with extracting and sequencing hundreds of millions to billions of DNA fragments is that you could detect as the ends of the fragments were usually damaged on a fragment. And so by being able to see this DNA damage, researchers had the confidence that they were actually sequencing ancient, authentic DNA and not contamination. So that was another huge leap forward. And so you would have to correct for this DNA damage in downstream analyses, but at least you knew you were working with ancient DNA and not contamination. So with these leaps forward and these advances in technology, researchers at the University of Copenhagen ended up publishing the first ancient human genome. And it was a individual from Greenland who lived 4,000 years ago and was part of the Sakak culture. And they sequenced the genome from hair from this individual that was preserved. And because they sequenced the near complete human genome, instead of calling it ancient DNA, they call it paleogenomics. And so in 2010, ancient DNA evolved into what is now known as the paleogenomics field. And so that was a very important point of the history of ancient DNA. Since then, the number of ancient DNA projects has just skyrocketed and increased dramatically, as is shown in this graph by Fox and Hawks. And also the sources for ancient DNA projects has also dramatically increased. But what has been lagging behind is thinking about the ethical implications of this research and ancient DNA research in general. And so what our lab tries to do is not only use these cutting edge technologies for ancient DNA research, but also we try to work to advance the ethics of paleogenomics. And I could talk more about that in the Q&A session. But right now I'm going to pass it on to Kelsey to talk about the 2 interesting projects.
Great, thanks, Ripan. So I'm really excited to share with you 2 of the projects that we've worked on at the IGB. And I think these projects both show how you can get DNA from some really surprising places, but also both of these projects were done working with communities to help them understand their history and learn from them as well. And so we had I'm going to be covering 2 projects today. The first is looking at DNA recovered from a pipe stem and using that to try and understand the ancestry of the owner of the pipe, who was an enslaved African, and provide that information for their descendants. And then the second project is actually using dog coprolites, which put simply is fossilized poop, and using that to understand the diet and health of dogs and the humans that they associated with. So to start with, the pipe stem that we worked with was recovered from a slave quarter at Belvoir Plantation in Maryland. And so this was done in collaboration with a local archaeologist, and this work is actually published in the Journal of Archaeological Science. And at Belvoir Plantation, there were enslaved people living there for decades. And so there are descendants who still live in the area, and they have a lot of interest in understanding where in Africa they come from. And so we were hoping to get some DNA from these clay pipes to better understand their origins and help them learn more about their history. And so we only had a fragment of a clay pipe, but this is what the whole thing might look like. And when I say we're extracting DNA from pipes, DNA can actually bind to some of the minerals in the pipes. And so that helps with the preservation. And so when we're extracting the DNA, we're actually pulling it back out of the clay. We're not just getting it off of the surface. And so this doesn't get us a whole genome. We got less than 0.5%, but that's still enough to kind of answer some big picture questions about ancestry. And so what we did first is we took the partial genome we had from this individual and compared them to a lot of worldwide populations. And so the pipestem is this the pipestem's owner, excuse me, is the triangle here. And they're most closely related to modern Africans. And so that's consistent with the fact that this was found in a slave quarter. And so we would expect their ancestry to be African. Something I want to note here, because we're going to be talking a little bit more about ancestry, is that ancestry is a little more complicated than we usually think about it. So I think often we think about ancestry as all of these different populations that have been the same through time, and they're kind of isolated from each other. And so when you say, you know, European ancestry, we sort of think of that as one group. But the reality is that, you know, through time, people move around, they interact with each other, and we know from history and also from a lot of genomic studies that just because we see, you know, this distribution of ancestry today, it doesn't mean that it was the same as it was 5,000 years ago. So I just want to keep that in mind as we move forward. And so knowing that this individual was most closely related to modern Africans, we decided to compare their genome to a lot of African populations along the western coast of Africa. And these populations were chosen mainly because a lot of the slave ships that ended up in Maryland came from the western African coast. And so on this plot, each of these dots or shapes represents an individual, and you can see that often the individuals within a population kind of cluster together. And the individual who owned the pipestem seems to be clustering with individuals from Sierra Leone. And so that suggests that their ancestry is most similar to modern day Sierra Leoneans. And so we were able to share with their descendants that that's probably where some of their roots come from. And so I think this is a really cool project, first because we really weren't expecting to get any DNA out of a clay pipe, let alone enough to kind of understand their ancestry, but it was also really neat to be able to share this information with their descendants who really couldn't get a good view of their history using other methods. So now I'm going to switch gears and talk about the coprolites, but first I want to give a little bit of context about where the samples come from. So these fossilized poop samples were recovered from a site near Cahokia, which was a large city that was the center of the civilization that we know as the Mississippians. And so the Mississippian civilization lasted for about 500 years. And as you can see from this map, it had a really broad distribution across a lot of the Midwest. And so the Mississippians had a very sophisticated and complex society. So we know they had complex religious rituals. We know that they had different classes. So they have wealthy elites and artisans. We know that they relied really heavily on corn agriculture. So that's kind of a common theme to what we see in Illinois today. And we also know that they had really broad trade networks. And so the Mississippian civilization was very influential during this period of time. And Cahokia was in a lot of ways like a modern city. It had a lot of cultures meeting and interacting. And just to compare, at its heyday around 1300 AD, it had a similar population size to London or Paris. So Cahokia was a big, bustling place. And you can actually still visit Cahokia if you would like. The Cahokia State Historic Site is in southern Illinois, close to St. Louis. And this picture on the right shows the kind of the central mound of the city that you can still climb up, and you can learn about the Mississippians there. So if you haven't been, when it's safe to go, I highly recommend that you do. And so there's a lot of interest about what life was like at Cahokia's beginnings, because there were people living there, but they had a very different kind of society. They lived in small groups. They weren't growing corn. And so we have a lot of questions about what their diet was like and kind of day-to-day life. And something that's helpful for us is that they had dogs. And so dogs are actually a really useful window into human life, as a lot of dog owners probably understand. Dogs and humans often eat the same things, they often move around together, they're often exposed to the same environments. So when we study dogs, we can get a little bit of understanding of what was going on in the humans as well. And unfortunately, we don't really have remains from this time period to kind of get that understanding, but we do have these coprolites, and coprolites can be surprisingly informative. So just to name a few things, first of all, coprolites can contain a lot of different fragments of scales or bones or seeds, and you can use these to identify the types of things they were eating. Another thing you can look at are stable isotopes. So a lot of you are probably familiar with carbon dating, which uses a radioactive isotope of carbon to figure out how old something is. But carbon and nitrogen as well also have stable isotopes, so atoms of different weights. And depending on what you eat, you get different amounts of those different weights of atoms. And so you can actually look at the isotopes to see, for example, if an individual was an herbivore or a carnivore, or if they were eating corn, which has kind of a unique signature compared to the other plants you can find in the area. And finally, and the part that I was most interested in, you can get DNA out of coprolites. And certainly a lot of it is contamination, but you can pull out a lot of other things too. So we can get DNA from the individual who made the coprolite. We can get DNA from the things they were eating. If they were infected with parasites or affected by bacteria, specifically diseases, we can pull those DNA sequences out as well. And finally, we can also get DNA from the bacteria that lived in their gut. And so this is actually a popular area of research in, modern humans, the gut microbiome, because it can be really informative about diet and health. And so taken together, just from a single coprolite, you can actually get a pretty holistic view of what was going on. And so at first we wanted to focus on diet. And so these were some of the species that we identified. You can see there are a lot of different fish and ducks as well. And so all of these are kind of, water resources, which makes a lot of sense because this archaeological site was right by the Mississippi River. So it makes sense that they would have been using that. And we also see a lot of different plants that they were eating, including walnuts and grapes. And finally, interestingly, we also find Toxocara. So Toxocara is an intestinal parasite that infects both humans and dogs, and it's often spread through contaminated water. And we also see, if we're looking for bacteria, we also find a couple of bacteria that kind of cause the same sorts of intestinal issues. And so that suggests to us that there may have been some kind of sanitation concerns at the site. And so just to kind of big picture summarize, what we can see is that these people at the founding of Cahokia ate a wide variety of fish and waterfowl and a lot of different plants. And even though corn was a really big part of their diet later on, at least at this time period, it doesn't really seem like it was a very big component. And then finally, based on the pathogens and parasites, we think that they may have had some sanitation concerns. So I hope that these two projects show the really cool things you can do with ancient DNA now that the technology has advanced. I quickly want to acknowledge a few people. I'd like to acknowledge the IGB and the collaborators on this project, and the other members of Ripan's lab, as well as the National Science Foundation for funding. And at this point, we're happy to start taking questions. Thank you.
Awesome. Thank you, Kelsey and Ripan. Like Kelsey said, we're ready to take questions, and we already have a number of great ones coming in. You can ask your questions directly at c2st2.cnf.io. So we'll go ahead and get started with some of the ones that got asked over the course of the program. The first one is, have you been able to continue to do lab work on ancient DNA over the course of the pandemic shutdowns? It seems like contamination is something you're already used to dealing with.
So Kelsey, maybe I'll go first and then you can answer. So the lab was shut down and the IGB was shut down from March up until about a month ago. And then a month ago, we were able to take, maybe I'll go first, certain precautions. So one person per lab, which is usually what we have in the ancient DNA lab, as well as another set of precautions to prevent COVID infection. And so we were shut down for a few months, but then about a month ago, we were able to open back up to a single researcher working in the lab. So.
I think all I have to add to that is, so really the lab work is just kind of a small part of what we do with ancient DNA, because once we get all of this DNA, then we have to sift through all the contamination and try and make sense of the data. And so all of that computational work can be kind of ongoing. And so even if the lab work part is stopped, we kind of have plenty of samples to work with to keep us busy.
Awesome. So that is actually a great transition for our next question. What are the ideal environments or conditions for DNA to survive in the remains that you end up sorting through?
I can take that one, Ripan.
Sure.
So in general, heat and humidity are really bad for DNA. So the perfect environment is probably something like permafrost, where it's very cold and it never melts. And that's also why a lot of ancient DNA studies are findings in caves, because it's very dry and kind of protected from the elements. So, I mean, you can get DNA from hot environments or humid environments, but in general, if you want really, really old samples to work with, you're only going to get DNA from them if they're from somewhere very cold and very dry.
Yeah, and I'd want to add to that we have been working with First Nations in British Columbia, and from ancestors from shell middens. There's something about shell middens that preserves DNA really well. We haven't done any kind of extensive analyses to figure out why that is, but it seems like some environment in these shell middens also really preserves DNA really well.
Awesome. And speaking of First Nations, one of our viewers wants to hear a bit more about ethical issues with paleogenomics and what interactions with First Nations or Indigenous folks look like.
So, I mean, it's really interesting right now, because usually for research with humans, you need IRB approval. So you have to go through IRB. People have to sign informed consents. But when you're dealing with someone who is dead, you no longer need IRB approval, even though the analysis of that ancestor or that ancient individual can strongly affect the linked First Nation or the linked Indigenous community. And so we are trying to make it a practice to where you have in-community engagement on ancient DNA analysis. So you work with linked communities on the project before you go ahead and do any kind of DNA analysis on that ancient individual.
And as a follow-up to that, it's a very related question, but someone was wondering how paleogenomics plays specifically into repatriation decisions sparked by NAGPRA or other laws like that.
So for NAGPRA, as far as I am aware, there hasn't really been a case where ancient DNA directly played into the repatriation and reburial of an individual. There is the ancient one in Washington, where a lab at the University of Copenhagen, along with other colleagues, did an analysis of the ancient one, and those results, I think, helped contribute to the repatriation and reburial of the ancient one. But as far as I know of right now, it hasn't directly played a role in repatriation.
Awesome. Sort of switching gears a bit, this question was asked of Kelsey. Referencing Schweitzer's T-Rex femur that was found to have soft tissue, we know that it is almost impossible for DNA, let alone cell structures and collagen, to survive a purported 65 million years. Do scientists have a mechanism that could accomplish this impossible task?
Do we think it's possible to get DNA from dinosaurs?
I think that's the gist of it, yeah.
I think that would be really neat, but I think we're a long way away from that, unfortunately. I think, so the oldest specimen that DNA has ever been gotten from is from a permafrost sample. It's from an ancient horse that was 600,000 years old. And then there's also a study, I think, of bacteria that's 1 million years old, but we kind of have to go back much further than that. I know there are some studies of soft tissue, and I know there's some study of proteins, And so it seems like proteins preserve a little better than DNA, and so that can push it back further. But I'm not sure we're going to see Jurassic Park anytime soon, unfortunately.
Maybe fortunately.
Well, yes. Depends on your point of view.
Yeah. Okay, so this next question is open to either of you. Have you been able to collaborate with people studying ancient languages? And if you have, any cool results?
So for ancient languages, there has been this language, the Dene connection between North America and Northeast East Asia. And so there has recently been some genomic results from an ancient DNA study led by Martin Sikora that suggests that there was also a genetic connection at around that same time period that might be connected to this language sharing. And so, yeah, there's lots of interesting questions out there to address. And I guess one point that I would like to make is that language and biology don't have to have the same history. You can have different language or different histories for language and biology, because you can have the spread of culture and language move and take over without the spread of people, right? So it's important to keep that in mind.
All right. This next one is also geared towards either of you. How are emerging sequences technologies, such as long-read sequencers, impacting aDNA research?
So I think, so specifically long-read sequencers don't really help us very much. So ancient DNA in general is very short. And so our goal is kind of how do we filter out the long reads, which are likely contamination. And so I think kind of a lot of the advancements in ancient DNA are kind of on either end. So in how do we more efficiently extract this ancient DNA, and then also once we actually get it back from the sequencer and we have this raw sequence data, how do we process it so that we can pick out as much of, the ancient DNA as possible? And so I think it's computational advances and figuring out better methods to get these tiny fragments out rather than necessarily the sequencing technology.
Yeah, I guess I would add one thing is that in some of the projects we're doing now, it's a comparison of ancient DNA from 2,000 years ago, 3,000 years ago, 6,000 years ago to living organisms. Maybe a salmon could be a good example. So with the ancient DNA that you can get from salmon, you're going to be limited to short fragments, and so you would use that technology. But with long-read technology, you can sequence genomes of salmon or any organism from fresh tissue in order to do that comparison between time.
And Kelsey, this next question is for you and goes back to one of your slides about Cahokia. But this person says, maybe the Cahokians just didn't feed maize to their dogs?
So, I mean, that's always possible. I think I would argue that most dog owners would agree that if a dog wants to eat something, it's kind of hard to stop them sometimes. But I do also want to make the point that coprolites, it's really hard to say, you can't say definitively that it wasn't in the sample, because what we're getting is a very small fraction of what, whatever they were eating. And also the coprolite reflects a meal. So it's possible these coprolites were made when, corn wasn't ready to be harvested. And so that's why it's not showing up. So I definitely don't want to say that it's, impossible that they were eating corn or that there's a difference between human and dog diet. It's really hard to kind of remove possibilities with coprolites. It's really just kind of the findings. So yeah, you could be right. So.
My dog eats whatever it wants, so I'm sure
Yeah.
Our diets look similar. This next question is opening things back up a bit, but are there any new potential technologies that either of you think would really help your research?
I would say that the analysis of proteins, ancient proteins, is looking really promising. because there are areas where proteins preserve better than DNA, and then also the results are more accurate. So we're definitely looking into heading down that direction.
I think I'd go with that as well.
Great. All right, this next one is a little fun. I heard that ground sloth poop in some caves could be cracked open and still be fresh, quote unquote, on the inside. Would it be possible to get DNA from this? Or is this even real?
There are studies that have gotten DNA from ground sloths. They didn't mention in the paper whether or not it was still soft on the inside. I know the samples I worked with were all very dry. I mean, they may as well have been chunks of dirt. But yeah, I think actually a lot of people have looked at ancient DNA of coprolites, especially for extinct species like the ground sloth and moas and things like that. So I think it's very possible.
I think it has.
And oh yeah. Yeah, we have just kind of our last question coming up that I think closes things up nicely. What is next for both of you research-wise? Are there any new projects you're excited about or new things you're working on in ancient DNA?
Kelsey, you want to go first?
Sure, yeah. So I'm actually working on a couple of projects looking at domestication. So a lot of them are about cattle. So looking at, so there are two species of cattle. There's kind of the cattle you see in America mostly, and then there are cattle you see in Asia, and they're different species, but they've intermixed a lot. And so there are a lot of questions about, when that happened and why that happened. And so I'm starting to explore that with some archaeologists to look at Chinese cattle. So that's what I'm working on right now.
Yeah, so we're actually heading in two directions. One is DNA sequencing technology has gotten even cheaper. So we can actually sequence human genomes to fairly decent coverage, like 5 times. And so we have a transect from these shellmans that I was discussing earlier, where the DNA is very well preserved, going back from 5,000 years to about 1,000 years. And so we're sequencing a transect of human genomes to see how the genome evolves over time. And so that'll be super interesting.
Well, that sounds incredible. I just want to close now by saying thank you to everybody for tuning in to today's program, and remind everyone that you can go to c2st2.cnf.io to evaluate today's program. That's the same place you were just asking questions, and you'll be entered for a chance to win a gift card. As a reminder, if you'd like to rewatch or you missed any part of today's program, this recording is going to remain on C2ST TV YouTube and our C2ST Facebook channel for as long as they exist. So please feel free to revisit this anytime. Thank you as well to Ripan and Kelsey for an excellent program, and to our partners, the Carl R. Woese Center for Genomic Biology and the Kleinman Center for Genomics in Business and Society. If you enjoyed today's program, please consider donating to C2ST at c2st.org, where you can also find archives of all of our previous programs, as well as timely STEM blogs. And just thank you again, everybody, for tuning in.
Thank you.